So this is session 2, day 2 of our 5-day review. And this is literally your test is a week from today. Remember, May the 9th is the day of that test. So you want to be ready, which means we will review also on that Monday before we, actually take the test. So we'll be right up to it. And please don't forget, you also have your test, your Unit 10 test tomorrow. So yesterday, what we should have left off on was the topic of we talked about the 4 major macromolecules. We said carbohydrates, proteins, lipids, and nucleic acids. I want to expand a little bit more on proteins, and we want to talk about something called an enzyme, which I might have mentioned a little bit yesterday, but let's get a little bit more particular. We said a protein that is specialized can become an enzyme, and enzymes are biological catalysts. And these biological catalysts basically work by speeding up chemical reactions and they do this by lowering activation energy. Now, activation energy is going to be described here in a second, but I want to kind of draw it for you. An enzyme has a very particular shape. Okay, and enzymes you will see. It's not an elephant foot. It's actually an enzyme. These different portions that connect to it are called substrates. And substrates are either going to come together or break apart based on the work of the enzyme. I should also mention this. These are also known as reactants, which are the left side of a chemical equation. These are the things that are literally being brought together or being torn apart to make a new substance. And we know, like, this little location where everything fits. Notice how it's got a precise shape. It's got a precise fit. We call that the active site. And basically, if we influence the size and shape of an active site, we can influence what can actually fit there. Maybe sometimes we inhibit things from fitting there, and that causes, maybe a problem, or maybe it's a good thing. But anyway, this whole little drawing that I did right here is an old-fashioned way, but still fairly relevant way, of looking at what we call the lock and key model. And just like a particular key can fit into a particular lock, this means they actually fit in place and work together. There's millions of keys out there, but only some of them will work in that lock and that door. Well, same thing's true of your enzymes. You have a lot of different enzymes in your body because you have a lot of different parts that have to fit inside of them. So we have a lot of variety there. We also mentioned this, like the enzyme is a protein, which means it's influenced by some of the same things. If we affect a protein in a bad way, we could basically be affecting its pH. We could be affecting its temperature, its concentration. All of these things will affect if a protein works. Think about it. Like if you cook meat, raw meat is very kind of stringy. It's tough. If you've ever had like beef jerky, it's really tough, right? Because the proteins haven't broken. They've just dried out. But if you look at somebody that like cooks a steak, they might put butter on there, or sometimes people will cook meat, under low temperatures over a long period of time. That is basically, we're destroying the proteins that make them up. So if we influence the temperature, or if we influence the salinity, or we influence the pH, we're going to break down those enzymes, which means they're not going to work the way they're supposed to. The other thing is when we think about this activity, that what they're actually doing, is if we look at a graph of them, and we'll draw everything in red here, that'll be no enzyme activity. We're basically going to have it go upwards, and then we're going to have it go downwards. Where I just put that X right there, that's going to represent the actual chemical reaction that's taking place. So we started off with, a certain amount of energy. Let's say that's a positive gain. That's a loss. Now, let's go ahead and change colors here. And let's go ahead and say we're going to have the same reaction take place, but with way less energy used at all. So that chemical reaction that takes place is going to save us that much money. So everything basically from here to here is what we call activation energy. And then this little savings here, that basically gives us another pile of money that we can spend elsewhere. I don't know, maybe doing the process again, which means we can do it more often. Ha. Now digestion just got a whole lot more simple because we just saved ourselves a lot of energy when we want to break things down, which is pretty common. Or we want to form something that saves us energy whenever we want to do that. This is basically the thing that influences how efficiently we can operate. They control, they cater how quickly these chemical processes occur. And once again, it creates the exact same product. It doesn't do anything less or more than what we are looking for. It does the exact same thing. So, with that said, do you guys have any questions on enzymes? No? Just a little aside, when we're talking about enzymes themselves, like enzymes fit together in particular ways, but we use something called an induced fit model now to really describe them. I don't think that word will even show up on your EOC, but there it is. And also, enzymes, when we say like their concentration can affect, how well they work, well, if you have a lot of an enzyme to break it down, that can help you out. A really good example of how various these things can be is, you've probably heard of something called lactose, right? Lactose. So if somebody's lactose intolerant, that means they can't digest the sugars found in milk. This tells you that it's a sugar because it ends with the letters OSE. But the thing that breaks this down, the enzyme that breaks it down, is called lactase. It ends with ASE, which is basically going to be an enzyme. So people who are lactose intolerant may be intolerant because they don't have the enzyme to break it down. This is also a lot of the reasoning behind some other digestive issues. You have an enzyme in your saliva called amylase, which helps break down really, really simple sugars in, like, if you've ever had a Ritz cracker and you put it on your tongue, it just melts. You don't even have to chew it. That's that enzyme at work. So it helps work on certain things, but not everything. Yes?
So the catalysts are the thing that speed up chemical reactions, right?
Yeah.
And then the enzyme is just like protein?
It's, in most cases, it's a protein, yeah. And, it does the same thing as a catalyst. It's just a biological catalyst. So it's like, there are chemical catalysts out there, too, that aren't biological.
So they're like the same thing almost?
Pretty much. It's just one they both facilitate chemical reactions occurring. Just the one basically uses a protein as that function. Any other questions? No? All right. So next topic, what we wanted to talk about was the basics of what cells are. Really the beginning of the study of biology. And this brings up the idea, whoa, that's terrible. Yeah. Yeah, it's actually Robert Hooke with an E at the end, right? He basically coined the phrase what a cell is. And we now today use it as the smallest unit of life. The smallest living thing is a single cell. If it's multicellular, that just means it's still a living thing, but it's made up of a lot of smaller living things. Robert Hooke came up with the first description of a cell. Another dude came along. His name was Antoine von Leeuwenhoek. He saw living cells crawling around doing stuff. All kinds of other people got interested and started researching what cells were. And basically, they all decided there was a theory that describes or supports the ideas of cells. And so there are three postulates of cell theory. And these three postulates are uniform. I don't care the order that you know them in. I just care that you know them. But the first one is just basically the definition of a cell. Cells are the basic unit of life. The second one is going to basically... Yeah, that's exactly it. Cells, there you go, come from other living cells. Now, this is a big deal because people used to think cells just spontaneously occurred, which we called spontaneous generation. We now have plenty of evidence that totally refutes all that thinking. But the other thing is, like, a dead cell doesn't give birth to another living cell. And the last and final point seems a little redundant. It seems like it's the same thing, but it's not quite. And it says all living things are made of cells. Or maybe if we want to be really picky, we could phrase it as all living things are made up of at least one cell, but whatever. So those are the three postulates of cell theory. Even though we've researched and we've discovered new kinds of cells out there, we've found out new components of them, this is always supported, and it's still supported today. It's just about as close to a biologic law as you can get, just because, well, it's pretty much an accepted fact. Anyway, we also, as we research these things, we notice there were differences between cells. They were all alive, but we noticed they were different, and there were two main categories. We called these eukaryotes and prokaryotes. Now, eukaryotes and prokaryotes, they actually used to have different names, but it doesn't matter now because we only use these two. Eukaryotes and prokaryotes have some major differences. The main thing for eukaryotes is that they have a nucleus. Prokaryotes do not have a nucleus. And that's actually where they get their name from. Eukary actually means like good kernel, which means like a good center, like brain thing. So that's kind of the leader of the cell. And then we have, yeah, actually we could say eukaryotes are usually what we call multicellular, or maybe single cellular, whereas prokaryotes are almost always single-celled, or unicellular. Also, eukaryotes have these things called membrane-bound organelles, whereas prokaryotes have no membrane-bound organelles. I'm going to shorthand it there because I'm lazy, but that's like the main differences between eukaryotes and prokaryotes. Now, they do, of course, have some similarities. They all have a cell membrane. They're all full of cytoplasm. They all have ribosomes and mitochondria. They kind of do their own thing, just in a weird way. Oh, I should also mention this. Eukaryotes are known for having much longer DNA, whereas prokaryotes have very short DNA. Prokaryotes, like good examples are like bacteria. Bacteria are very, very, very simple. And they don't have a whole lot of genes they need to make copies of. So that's why they reproduce so fast. They don't have organelles they have to like make a separate pile of, and then they have to divide later on. It's not what they need to do. So they can reproduce really quick. Eukaryotes, they take more time. They're more deliberate. They have a much more precise way of dividing themselves and copying themselves. And we studied that pretty extensively, which we'll get to later. So those are the main differences between eukaryotes and prokaryotes and the basic ideas of cell theory. Anybody have any questions there? No?
Oh, wait.
Yeah.
That one theory with that, or whatever, or like, I can't remember which way it went, or it's like eukaryotes like create prokaryotes, or like vice versa.
Oh, yeah. It was prokaryotes formed together to make eukaryotes. We called that endosymbiotic theory, which we'll get to later because that kind of fits in with origin of life. You know, how did life get more complex? That's really one of the leading thoughts on it. So we'll save that for later, but that's a good thing to remember. So let's go ahead and try looking at these eukaryotes again. There's a U in there. Now, eukaryotes can be broken down into two main types of cells. We have animal cells, and we have plant cells. Now, plant and animal cells are kind of unique, but they do have a lot in common. So I'm making like a Venn diagram here. Squares are easier than circles. Anyway, so what do they both share in common in terms of what's membrane-bound organelles they have? Well, there's a few obvious ones. Of course, they share a cell membrane, which we'll talk about on the next slide in better detail, or actually after that. We also know that they both have a nucleus, a nucleolus. We also know they have ribosomes. They have a rough and smooth endoplasmic reticulum, or ER. We also know they have a Golgi apparatus or complex. I'm going to say complex because it's got the letter X in it and it looks cooler. We also know they have things like vacuoles. They have vesicles. I should spell it right. They also have cytoplasm and mitochondria. Say that one more time. Powerhouse to the cell. Thanks for the delay in when I asked you and when you answered, because it made it seem like you didn't say anything. It's just funny. That's good. That's good comedy right there, Sean. You should hit the stage, man. I want to see you up there at the Apollo. But anyway, animal cells have a few unique features. They only have mitochondria, which Sean pointed out for us is the powerhouse of the cell. And that's because animals only take in the stuff for cell respiration. That's what mitochondria does. That's how they generate their energy. We also know they only have a cell membrane. We also know they have like various and smaller vacuoles. And yeah, those are kind of the main differences for animal cells. For plants, though, they have chloroplasts. Now, I'm not saying chloroplasts replace mitochondria. Notice on our in-between area, we actually said both of them have mitochondria. So plants also have mitochondria. I don't know how many times kids have told me this, and they say something like, well, only plants have mitochondria. I'm sorry, only plants have chloroplasts and only animals have mitochondria. Not true, because cell respiration also happens in plants. If I were writing the test, if I were writing the EOC, that would be a question I would put in there, because it really tests your knowledge if you understand that there are similarities between plants and animals and what parts are actually working there. I should also mention, since we said chloroplast, they have a little component called chlorophyll. And chlorophyll is really important because basically it's the light-absorbing molecule in the chloroplast for photosynthesis. We also know that these things have cell walls. So they do have a cell membrane. That's also not replaced. But it's a tough exterior to the cell that gives structural support. And also, speaking of structural support, they have a large, centralized vacuole. And this vacuole, you know, in animals, the vacuole contains a lot of waste products. But when it comes to the actual plant, it's got like a giant swimming pool on the inside, and it fills up with water. And it's like a giant water balloon that's really adding extra pressure, pushing out of the cell, which makes it tougher to squeeze, which means it's more strong. This is why trees can be super heavy, because you know trees are stupid heavy, but they're also really tall. It's because they can support their own weight, even though they don't have bones. So that's like a major difference between plant and animal cells. Another thing kids always bring up is that, oh, animals are eukaryotes and plants are prokaryotes because they both start with P. No, they're both eukaryotes. You should know that. Look, I even wrote eukaryotes up here. It's there to tell you. They're both eukaryotes. There's also fungi, which are more like plant cells. They have a different kind of cell wall. And there's protists, which are just weird and may be more like animal cells, but sometimes more like plant cells. I don't know. So, yeah. Doing okay there?
Yes.
Any questions on that? Yes, sir?
Which one's, so between vesicles and vacuoles, which one is the vehicle?
The vehicle one is a vesicle.
Okay.
Yeah. And actually, you're kind of bringing up a thing here. I'm not going to go ahead and define what every single one of these things do. I'll highlight a few of the key ones coming up, but like, yeah, you should kind of brush up on what the functions are for those different organelles. Which now brings us to this thing. Most of the cell I'm going to use as the inside of a cell. Okay. Okay. That's a cell membrane. So inside of here, we're going to ask the question, how proteins are made. And this is like that sequence of where they visit. So we're going to start off the inside here, drawing everybody's favorite. That's terrible. Yeah, it's the nucleus. Inside of the nucleus, there's a more dense region here that's called the nucleolus. And then inside of the nucleolus, we make these things. Let's make them purple. That'll be nice. We make these things, these dots, and these are called ribosomes. Now, when we go back, we think about the design. The nucleus has a nuclear envelope which basically connects to this rough ER that it in turn is attached to this smooth ER. Yes, that's the main difference, is that the rough ER is known as being bumpy. So it has these purple things, these ribosomes, stuck on the outside. That's what it does. And what do ribosomes do? Their whole purpose is they make protein. Now, when we think about those ribosomes doing that job, let's go ahead and make this red. We're going to see a whole bunch of proteins that are basically going to get made kind of randomly, and they're eventually going to move to this region. And they'll kind of get sorted into the little ends here, and this makes it easier to transport them later on. I should also mention this area that we're playing with right there. This is this. We've got the rough ER and the smooth. Notice there's no purple dots, right? So there's no ribosomes on the outside. It actually does have another purpose. It makes lipids, which are really important in your cell membrane and like all these other membranes. But it basically sorts these things out through these little vehicles, these little vesicles. And they usually are filled with our little enzymes, right? And then they get transported to another region, which we like to draw kind of like this. And that's called the Golgi complex or apparatus. And we said when you think of the Golgi complex, you want to think of FedEx, because basically all it does is it sorts and organizes these proteins based on what they are so they can get easily transported elsewhere, probably out of the cell. So, go ahead. Kind of. The smooth ER kind of just groups it by like a category and just sends it off. But where a Golgi complex literally sorts each individual one out and says, there are 7 here, there's 4,000 there, and I need 600. So it'll pull it from the proper area and send out exactly how much it needs. Woo! That's recorded now. So, you know, we'll break off some more vesicles, and they'll basically go and they'll merge with the outside of the cell membrane. And what we'll see is what happens to those proteins is that they get transported, hopefully, to exactly where they need to go outside of the cell, and they escape. So they're expelled. That is basically the story of protein. Now, the truth is, these ribosomes, we said they're pretty important, but here in the nucleus, we had a process that we'll bring up later in the week. We have this thing called transcription. And then we said in these ribosomes, basically what's going to happen here is going to be something called translation. And that's actually where we assemble the protein. So it gets even more specific, more detailed in there. But anyway, moving on. Can I do that? Yeah? Any questions? All right. So I also want to take the moment now to talk about that really important cell membrane. The cell membrane is wildly important because of the structures that make it up. First off, you need to know what a phospholipid is. phospholipid is basically, it's got a head, a round head, and two wiggly tails. Now the head is what we call a hydrophilic head. And you're right, it does like water, so it's got a happy face for water. Loves water. Whereas the tail is what we call hydrophobic. And I'm going to draw a frowny face. It doesn't like water. It's actually afraid of it. It hates it. So the phospholipid design makes up most of your cell membrane, but the truth, no, maybe not most, but well, anyway, the point is, it can't just be one layer of these things. That's why we use this phrasing called a phospholipid. Yep, a phospholipid bilayer, which kind of looks like this. That's a goofy looking protein. So is that one. Wiggles. All wiggles. Now the truth is they're not really this spaced out, but you know, I'm just trying to give you a general idea here. There's also maybe a few things plugged in there. There could be some like cool little extender thingies here. So we've got two layers of phospholipids because remember, on the inside and outside of the cell, we need water to be able to touch it because there's water outside of your cells as well as inside. But also it prevents water from moving through the membrane because there's that really bad area that the water doesn't like. Also, there's these big guys. These are proteins. Man, they just keep showing up today. Proteins that act as like channel proteins or transport proteins or integral proteins. That also is a protein. We also have this thing in here, which I'm basically going to draw that as cholesterol. This is the good form of cholesterol. It helps strengthen your cell membrane. And then we have these cool little receiver guys on the outside. These are essentially carbohydrates, sugars, that kind of act as a communication method in the membrane. When you look at this, it tells you there's way more than just a phospholipid bilayer. There's a layer of all kinds of things in there, which we use a phrase to describe that, and that's what we call the fluid mosaic model. And that describes all the different pieces and how it can move around and kind of wiggle. That's really important for us because it's basically going to make sure that there's enough pieces and parts that can do all the different functions and demands of the cell membrane. We should also take this as an opportunity to kind of talk about how things can move in and out of cells, which kind of now brings up the idea that we need to describe what that stuff is. And basically, we have this phrase that we use to describe the cell and how it functions called semi-permeable, sometimes called selectively permeable. Okay, and semi-permeable and selectively permeable is basically going to be allows some, but not all, things to pass through. Like I mentioned earlier, water can't freely pass through here necessarily, so there's got to be something in place to help it move along. Another important thing you want to remember is we want to like kind of use proper terminology here. So it brings up the idea of solutions. Because pretty much everything is getting moved around in a form of a solution. You don't just take a salt granule and just push it through your skin. You know, we dissolve that in water. That's called a solution. So we have two parts of solutions. We have things called solutes, and we have things called solvents. Solutes are found in the lower quantity, and they get dissolved. Whereas solvents are higher in quantity, higher in amount, and they do the dissolving. Okay? So the basic idea is that everything that is an aqueous solution usually has water as the solvent, and all the other stuff that's floating around is called a solute. Describing what solutes are helps us describe their concentration, and in turn helps us describe how things actually move through the cell membrane, which we call cell transport. But how are we doing here? Any questions? No? All right, so let's talk about cell transport. Go ahead. Gives me an excuse to drink some water. So I have 13 minutes. Can I do cell transport? Yeah, I think so. You're so cool, Sean. Am I good, Cam? All right. So, looking at cell transport, we get reminded of our best friend. No. The triangle of love. Whatever. He has many names. So, if we go down the concentration gradient, we go from a high point to a low point, we call this passive. But if we go from a low point to a high point, we call this active. This describes the forms of transport, okay? When you move up a concentration gradient or down a concentration gradient. By concentration gradient, I mean how much solute there is and how not much solute there is, or it could be solvents too. The first form of this is called passive. And the main forms of passive transport that matter to us are going to be the first one called diffusion. And diffusion is basically the movement of solutes from high concentration to low concentration. Okay? To draw this, and hopefully a pretty good context, here we go. It's macaroni time, y'all. Right there, I just drew a cell membrane that's semi-permeable. And on both sides of these things, I'm going to have an amount of water. And on one side of that, I'm going to put a ton of a solute. Let's say it's sugar. Why not? And on the other side, I'm not going to have very much sugar at all. By nature, by diffusion, the solute wants to move from a higher concentration to a lower concentration. Okay? That's what diffusion basically is. But there's other processes that are also passive. A really good example is something called osmosis. Osmosis is basically the diffusion of water molecules. Now, when we look at the same example that we have here, we can actually see that both can actually occur at the same time. So just by looking at the right side of the tube here, we have very, very few sugar molecules, which actually tells us we have tons and tons and tons of water molecules because they're not being occupied by the sugar. So that space, we have the same amount of water on both sides. The water on the right side is going to be in higher quantity. When we look on the left side, there's less space for water because there's more sugar. So that means there's less water. Okay. Which means the water wants to move from this direction to this direction. That's what diffusion is. And by the way, this will cause the water levels to change. That means water will rush to the left side and cause the water level to rise, and that means the water level will drop on the right side. This means diffusion and osmosis can happen at the same time and move in opposite directions, which means you can actually predict how cells are going to look after a period of time. Also, I should mention, another fancy version of passive transport is something called facilitated diffusion. And facilitated diffusion means a protein channel helps move the solute. It's the exact same thing as diffusion, but they just need another tube to travel through. It doesn't work against the concentration gradient. It goes down the concentration gradient. And basically, the main thing that should jump out to you is when we think of passive, that means no energy is used. Everything we just looked at here, osmosis, diffusion, facilitated diffusion, all of those do not require energy. It's free for our bodies to do. It'd really take up a lot of energy if we had to wait, to build up energy to move water into our cells. That would be bad. So the other version of cell transport is called active transport. And active transport comes in a few flavors as well. We have something called endocytosis, something called exocytosis. Now, endocytosis, the keyword endo means into, which basically means we're going to move things into the cell. It comes in two varieties as well. One's called pinocytosis. The other one's called phagocytosis. Exocytosis is just exocytosis, but exo means exit or leaving. So it leaves the cell. The stuff that we're trying to get rid of actually just passes throughout. Pinocytosis is basically stands for cell drinking. So it's when a cell takes in very small particles, very, very, very small particles. Just like if you're drinking something, you don't drink a steak because it's a lot of particles all at once. Phagocytosis literally means cell eating, which means it takes in larger molecules. If you remember when we talked about the immune system, we even mentioned things called phagocytes, which are basically large white blood cells that eat and digest other cells, literally like bacteria cells, because they're gigantic. So they literally surround it and wrap it up and enclose it, and then after it's engulfed, breaks it down. Exocytosis, there's nothing too fancy about it. It just is basically cell pooping or cell throwing up, and it just expels, just expends all that stuff, just gets rid of it, shoots it out. And that's the basic concept there. Remember, active transport uses energy, which now brings up the idea of what form of energy does it use? Well, there's something out there called ATP, adenosine triphosphate. Tri means three. So ATP has three things called phosphates, three little dots. And this forms a relationship with another molecule called ADP, adenosine diphosphate, which means two. Now, ATP is known as the high energy molecule. Okay? And it's the most common form of energy we use in our cells. If we want to change that, if we want to release that energy, we basically shoot out those phosphates and we end up with ADP. It's the exact same molecule, it's just missing a part. If we add in that part, we end up recreating ATP. So ADP is a low energy molecule, ATP is a high energy molecule. So the other question is, how do bodies or how do organisms find a way to make this energy? We're expelling it, we're gaining it back, but how do we make that energy happen? And that all goes back to the idea of cell respiration and in turn photosynthesis. So let's do that real quick, like as short as I can possibly make it. Photosynthesis is a pretty simple formula. You guys should know it by heart. First off, what do plants need to survive? Well, they need water. They also need carbon dioxide. They also need sunlight, but I'm going to write it here on this reaction line. Really, it does kind of belong over here, but it's not like a chemical, so I can't really put it into a chemical equation. Anyway, then it makes, as a byproduct, oxygen gas and something called C6H12O6, which you guys remember is our sugars. It's glucose. The truth is plants really don't make glucose. They more so make a starch, but whatever. It's functionally pretty much the same thing. There's another process called cell respiration. I should also mention this is called aerobic cell respiration because it uses oxygen, which means they take in oxygen, they take in sugar, and they react and they make water as a byproduct, CO2 as a byproduct, and energy. But now this is an actual chemical form of energy that we like to call ATP. That's what that process is. You will notice that everything on the left-hand side here, we call these the reactants. Everything on the right-hand side here, we call these the products. Notice the relationship between photosynthesis and cell respiration is the idea that the reactants of one become the products of the other. The products of one become the reactants of the other. That's the main concept that you need to be able to follow and look at. I don't know how many times I've ever seen people propose questions about, explain this cool thing about photosynthesis. A lot of the times, like maybe on the EOC, they'll ask you this. They might ask you, where does that carbon come from? It's a really good question. Did it come from the water? No, because there's no carbon involved. It had to come from the carbon dioxide, which tells you part of the sugar that you eat came from the air that you breathe out, which is pretty insane. Also try to keep in mind, this sun is a form of solar energy, and then we have this ATP is a form of chemical energy. And that basically is created from the breakdown of the sugars, because sugars store energy. We have aerobic cell respiration, and tomorrow, actually not tomorrow, on Thursday, because you have your test tomorrow, there's another form called anaerobic respiration, which we'll talk about as well, and that's where we'll pick up.